Happy to. How are you this week? Now, I'm sure you are getting a little bit sick of me this week, and I decided to skip my face in many of the video this week, and you are finally seeing me again this week to wrap up for all the lectures that you have been watching. Okay, and today's lecture is going to be a little bit shorter, so the contents are not that intense in my opinion. We're going to pick up what we talked about on Tuesday's lecture, and then I will also give out instructions on assignments. So very important. At the end of this lecture will be instructions for this week's assignment, so you have to watch the whole thing. Now, in case you're wondering what I'm wearing my lab jacket. Yes, I kept my lab jacket home for a while now in case things happen. So my son and I have been interested in doing experiments, and he has his own lab coat, and I have to bring my own lab coat home from school. So that is what's going on. Now, so before we go to lecture, let's take a look of my lab and see some of the common things that are used in biotechnology. All right. Alright guys, what you just saw in that short 20 seconds clip was the lab, was my lab, okay. So to clarify, I shot that video during spring break, okay, before everything crazy happened. So I just want to make sure, like, oh, did Dr. Hong went to the lab, okay, this past week? No, okay, I haven't been to school as long, for the whole time that since the closure, okay. Well, since that one Tuesday I shot one of the video, but anyway, I didn't go to the lab for the last 2 weeks, okay. I'm not that crazy scientist yet. Okay, so today we are going to pick up from where we left off from last time. Last time we were talking about recombinant technology, having to how to put in a specific gene, okay, into a plasmid, and then put into a little organisms, and then have the organism secrete the protein, okay, the enzyme or antibody that for therapeutic use. Okay, now here we talked about last time how to screen for those using part of the constructions of the plasmid, i.e. having some type of an ampicillin resistant gene, or having some LAC-Z gene to do the screening for successful cloning. But the real screening people need to do for good verifications is actually to do a PCR, or polymerase chain reactions actions there. And we're going to talk about briefly, okay, some of the concepts behind. We're not going to focus on the technical details of how to do one of those things. Okay, it's never going to be technical. It's more of an informational background. Okay, and here looking at this little left hand corner graph here, we have the it's a Agarose Gel Image, okay, Agarose Agarose Gel Image, and inside here, these little pieces, these are DNA, okay, double-stranded DNA. And in fact, this little gel image, it's from my lab, okay, student produced that, and the idea, okay, if you apply imagining this gel and in association with the plasmid, is that when you have a plasmid, alright, with the gene being inserted into the plasmid, you can do PCR, okay, to verify this segment being present. If yes, there will be a band. If you have a construct without the gene, okay, a strict line, then no construct, then there will be no band appear in this agarose gel. So this is a very good way to verify the presence of your inserted genetic material into a plasmid before you put before you start putting it into your organisms and having it produced. If you don't do this verification step, you ended up not having any proteins, so you waste a lot of downstream time. Now, I personally don't do a lot of verifications in my step. Maybe I should. So because of the success rate of these insertions are not that high, but there's time and cost factoring into it. So if you're really doing lab, these are very crucial factors people need to think about, not only in academic lab but also in industrial labs as well. Right, so here is some of the general topics for the next three weeks. I know a lot of students don't like to be living in the dark. You don't know what to expect for the next 3 weeks. Okay, I've been kind of vague. Okay, even the syllabus, we're saying that the original syllabus schedule saying open topics. Now here are the topics. Now still subject to minor changes potentially, but here you get a bulk of an idea. For the next week, we are going to talk about antibody usage in diagnostic and biosensing, and these topics are very topical, indeed related to COVID-19 diagnosis. I try to make it relevant. All right, we'll talk about some of the assays, including ELISAs, lateral flow assays or dipsticks, flow cytometries, and many more, and to tell you how antibodies are being used to facilitate diagnosis. Now, in today's topic, we're talking about nucleic acid diagnosis, right? We're doing PCR, okay, to generate genes, okay, to see if that organism presence. But there are many other ways to do diagnosis. Okay, we're going to talk about it so that and potentially the assignment is also related to what we're talking about, so it will still be related, revolving around COVID-19 diagnosis topic there. Okay, and then the following week, week of 4/13, after you guys come back from the Easter holiday, we will talk about antibody usage as a drug conjugate. Okay, we've been looking at antibody as a therapeutic agent by itself in the JAM modules, and we touch upon a little bit in this class as well. Okay, it directly bind to a certain target receptors or interleukin, things like that, to block the signaling pathway. All right, what about it is linked to a drug? Okay, what else you can do? And we've been talking about the end result. Okay, in this biotechnology class, we'll talk about the concepts behind identifying, meaning screening these particular antibody, characterizing these antibodies productions, and as well as the applications in fields such as cancer. And we have a specific drug that is many more now. One of the earlier one is a antibody that is specific for breast cancer and conjugated to a drug. Okay, so there's many more now, but we will pick some as a talk and considerations behind it, and potentially advantages and hurdles. Okay, now these topics I as a graduate student, some years ago, I spent a long time investigating how antibody drug conjugate works, and wrote a whole proposal on it. Didn't get to do it because of money. Again, out of money. My advisor out of money. Like, you're stopping that. But anyway, so that I consider myself a half expert in that area, at least a theoretical expert in that area. And then the last week of this biotechnology class, we are going to look at a group of molecules called aptamer that is, I consider as a molecules beyond antibody. All right, so this is really my life work is in so far. My lives of research work is in the entire dissertations on that area, and I'll use that little lecture or two to talk about that in terms of its background, identifications, and application. Again, it's not going to be technical detail. It's more of a broad sense so that you're aware. So at least we have one therapeutic agent called macugens are approved for treatments of age-related macular degeneration. Right, we talked about it in the self-care. right? Quite a long time ago, it feels like. And this is belong to an aptamer, okay? And then we'll look at some of the investigational effort of pushing aptamer as a therapeutic agent, particularly in areas of thrombotic events, okay? We are talking about clotting, okay? We're clotting, so, okay. And there are currently I'm also working on areas that are related to clotting as well, and there are a few students in the pharmacy school that are involving in that research. And then lastly, the last class, April 28th, we'll do a wrap up of whatever is left over to be wrapped up, and most importantly, we will talk about final exam details. I know you guys been wondering what is going to happen with the final for this class. At this point in time, when I'm recording this lecture, I have no idea yet, but I can assure you it's not going to be something crazy. Alright, it's not going to be something crazy. Alright, I know you have many other things going on, and then you have the GI module going on. Everything's online. Think life are changing rapidly. So I completely understand. So I don't want to create a lot of extra unnecessary stress on you guys. Okay, so be sure it's not going to be something crazy. Right, so today's learning objectives, okay, after almost 10 minutes of small talk, now it finally goes to the objective. Today's lecture is not going to be very long, okay. We're going to talk about some principles behind PCR and RT-PCR and understand how PCR, RT-PCR work as a diagnostic tools. And then the last objective is tied into your assignment for this week, okay, is to look for some PCR technology on COVID-19 diagnosis. Again, very topical type of topics, all right. Okay, so how do we know if you have the right plasmid? All right, so that was the introduction that I gave. If you have a plasmid that containing the right gene, okay, what can you do? You can do a experiment or a process. Okay, that is a very widely used process now. Almost all molecular biology related lab are doing these type of reactions. Okay, hospitals, lab are doing these too. It's called polymerase chain reactions, commonly abbreviated as PCR. Okay, you may have learned about it some time ago. If you have a more rich background, okay, such as if you have a bachelor degree, you're probably quite familiar with this term. So it is an in vitro technique, okay, and it amplified a small amount of specific DNA sequence and used for further testing and verifications. In our case, in our introduction, is to verify our inserted gene. Okay, is it there? All right, if yes, it's there, it's going to be able to produce the protein, i.e. it could be the antibody. Okay, it could be the antibody. It could be the enzyme. Okay. So all these biologics that we touch upon in the modules. Okay, so this is one of the probably most straightforward steps to do this verifications. All right, a little bit short history of PCR. You're not going to memorize anything like this. Now, PCR was developed not too long ago, actually less than 40 years ago, okay, about 37 years, by Dr. Kerry Mullis. Okay, this little gentleman here, he actually died last year sometime, so he developed the PCR actually while he was still a PhD student, right? He was that amazing. And then following the timeline there's publications of PCR and then he formed joined corporations, and then it published in Science, okay, Science and Nature, or those elite science research journal that everyone wants to publish, but it's super competitive to publish. I don't see myself, unfortunately, I don't see myself publishing in Science ever. That is a sad truth. Anyway, and then further down, and there is applications, okay, using Tap Polymerase in PCR reactions, and along the line there is the inventions of automated thermocycler. So before 1988, okay, between the 5 years time, people are doing very old school reactions. Okay, we're going to talk about it a little bit. I've never seen it. I heard of it from a professor when I was in undergraduate and a long time ago, and he was an old guy, and he was mentioning how he did PCR reactions back in the days. Right, back in the days. Anyway, and then we'll and this tag polymerase, we're going to talk about it, and it's being named as or declared as molecule of the science of the year in the science back in 1989, like still long before you guys were born. Okay, long before you guys were born. And then further down there is a modifications or small incremental improvement in the reactions using fluorescent DNA dye. Okay, to was used. Sorry, there's a typo there. Was used. Okay, apologize for the typo. And your versions, I will make sure your versions will be corrected. Okay, the student versions will be corrected. Used to monitor DNA amplification, so-called real-time, in a sense, lay the pathway for real-time PCR. And there's further down, we have the reverse transcription PCR. Okay, this technology really helped to diagnose for RNA virus. All right. The most discussed RNA virus these days is our COVID-19. Okay, it's our COVID-19 or the SARS, version 2, whatever name that they call it, right? And then in 1993, after 10 years of inventions of PCR, Dr. Mullis was awarded with Nobel Prize in chemistry, and it is really a huge thing. This technique, benefits so much, and the impact is tremendous. It's tremendous, tremendous impact. Because I do it every day, almost, when I was in school. Okay, so it is time to cook. And why I say it is time to cook is when you do a PCR reaction, it is almost like baking. It's almost like baking because you need a lot of ingredients being put in there. You need so here it's some of those ingredients. Well, you need to amplify a specific DNA. Of course, you need to start with a DNA, and then you need your building blocks, your nucleotides, ATGC. You need a tube, okay, as a container, and you need some conditions like a buffer to, you cannot do a reaction dry, right? And then here is another one. It's called primer. Okay, primer is where is a starting point for our enzyme, to start to work. It's the tag polymerase. Okay, tag polymerase is the enzyme. So these are the basic ingredients to perform a PCR reactions. Now the principles behind PCR reactions is really a changing of temperature. From high, usually we're talking about 95 degrees Celsius to 100 degrees Celsius, to a little bit lower temperature, about 55. Now you don't need to memorize these degrees Celsius, and then and then cycling back to a medium-high temperature of 72 degrees Celsius for the reactions to happen. Like the basic one cycle type of a breakdown is you start with a double-stranded DNA. Now think back to the in vivo process you learned in biochemistry. Right, there's something called the helicase, kind of unwind it, and then you have a polymerase to go in, and then you extend, right? So in a tube, we are not having any type of helicase or something to unwind your DNA. But the DNA is very interesting. You can heat it up, and the two strand will break apart. Okay, will break apart. Basically, you're having hydrogen bond between the two strand there. It will break apart, but this breaking is reversible. It's reversible. As long as the temperature goes back down from 95 degrees Celsius, it'll come back together. You're not gonna cook the DNA essentially at 95 degrees Celsius. Anything higher than that, yes, you can potentially cook or totally destroy the DNA. But by heating the DNA in solutions at 95 degrees Celsius, you're not gonna permanently denature or breaking it. It will come back to the original form once the temperature goes down. So you're breaking it apart so that it create a gap. Okay, create a gap so that the polymerase can start. but you need some starting point for polymerase to recognize. So that's why we give some primer, so which is the starting point for the reactions. Now you lower the temperature a little bit to 55. At that temperature, the larger strand, the two strand, won't come back together yet, but the smaller pieces, primer, it's a good temperature for them to come back together. So they annuled into a partial double-stranded regions right here, and then at this point, the temperature to go up a little bit higher, just enough for them to not dissociate, and at that point, your Tap polymerase will be able to do the job for extensions, incorporating the nucleotide, and then one by one ATGC, and then goes on. And then you, after one cycle, you start from one copy, become two copy, and then these reactions are exponential. It's an exponential growth. Alright, so you go from 1 to 2, and then 3 cycles, you get 4, and then exponential growth into copy that you would like. So you still, ended up, after a couple of cycles, you have your original copy. So it's an exponential growth. 1 cycle, you have 2 copies. 2 cycles, you have 4 CYCLE cycle copies. And then 3 cycles, you have 2, 4, 4. That's doubling the 4. Right? It's 16, 2, 4, 16, 32. All right. So and so on and so forth. So if I were to ask you how many copies you have after 5 cycles, right? It'd be 64. Now I don't know if I asked you these type of question, but just get an idea in knowing it is exponential growth. Alright, so here is a graph to show you the visual changes in terms of the temperature. So I go from high, 94, 95, and then go down and go up again. So this is one cycle here, so it's a 95, 55 or 45 here sets, and 72, that's one cycle, and this is the second cycles, and so on and so forth. And here is a picture of a typical thermocycler that is being used in our lab these days. Now, I talked about in the old days before this inventions, what are you going to do? We want to change temperature rapidly. So basically, you would have 3 water baths maintained at 95 degrees Celsius and 55 degrees Celsius and 72 degrees Celsius. Basically, people would manually put the tube inside each of those and then time it with a timer. Alright, and then come back up. For example, 1 minute here, 5 minute here, 7 minute here, whatever. I'm just making up the time. Alright, and then come back to this cycle. So it is very tedious work. Before this invention, thermocycler inventions, people need to dip their tube into bath. And of course, when you're transferring from one to the other one, chances are it takes time to go up temperature or cool down, and it takes way longer for these reactions to complete, and you're not having a precise control, and ended up, if you do a automated cycle, machine cycle, maybe 5 cycles, you get enough copy of DNA that you would be able to identify or image it in the gel. In a manual steps, perhaps it's gonna be doubled. It's probably gonna be doubled in many cases. These are just FYI small details. Alright, and then we're going to talk about the enzyme that is most important for this reaction. Now, this is the first one, Tap polymerase. There are other polymerase utilized in the PCR reactions these days, but this has been the first one, so we'll talk about it. Now, Tap polymerase is thermostable. Looking back the reactions, the mixture heat up to 95 and cool down and heat up. Normally any proteins will get destroyed with of these type of temperature changes. Think about the egg. Okay, you put your boiled egg in 95, it boils, it come back up, it's not gonna go back into liquid form, right? It's a one-way step. But on the other hand, Tap polymerase is not going to be destroyed by high temperature. All right. So this Tap polymerase is named after a organism, Thermus aquaticus, all right, that live in hot springs. Okay, so they are they evolved into a way to withstand such high temperature. And now these Tap polymerase are being commercialized in a small tube here. Now, talking about commercializing Tap polymerase, I have a small story to talk to you guys. So I told you, in my lab doing PCR reactions, it's a daily routine, especially in the summer. Everyone is doing PCR, and so I do a lot. I'm thinking perhaps my lab uses the most amount of Tap polymerase in the entire Northeast PA region. Yes, I do use a lot. Now, these commercial tube, for a tube like this size that in the figure, it range about $120 or so, depending on what brand you're getting. Okay, so it's quite expensive, and this particular one can do about let me think, about 100 reactions or so, 100 reactions. So it's not very it's not a lot, and typically for my weekly usage is many reactions. So if I were to use something that I need to buy every time, I'm gonna run out of money very quick. And in my lab, we do produce Tap polymerase. So it is now produced in with recombinant DNA technology that we talked about. So basically, I have a little E. coli in my lab, sitting right now in a deep freezer, negative 80 degrees C freezer now, and they have their genomic DNA and they have a little plasmid DNA which will produce Tap polymerase for me. All right, so we students in my lab just did that last summer, the summer of 2019, and the amounts that we produce, I think it's a commercial value. I think it's a 5-figure commercial values there, 5 figures. But we're not gonna sell these Tap polymerase. So these, I'm just saying to the message here is, doing recombinant protein production these days are not something that is beyond imagination. It's happening right here, in any moment, right here at Wilkes University. Okay, at least in my lab. Right. So what can you use for to use PCR beyond checking your plasmid having a right set of a gene in there, is to use it as a molecular diagnostic tools. Okay, the reasoning behind is to detect a low amount of specific pathogens with a known DNA sequence that is unique to that organism. Okay, this could be bacteria and viruses, and other usage could be including in genotyping certain cancer type. Are you expressing that certain receptors? All right, remember receptors, and going back, it's coded by the gene. Okay, coded by the gene. Are you expressing that gene that code for that receptor? So these techniques are very useful. Now, particularly these days, we are talking about, hey, very shortly after we see this virus causing the COVID-19, we know the complete genomic sequences. Okay, then people can look at some of the unique regions in that virus, and then start going through the process, synthesizing a basically looking for some primary regions so that it can bracket certain gene and be able to tell if a samples contain a particular virus. Now here we step back, work a little bit, looking at influenza A. Okay, flu viruses has been around for very long time. And one of the technique to diagnose influencers is diagnosed with reverse transcription polymerase chain reactions, or RT-PCR. So it's a little bit advanced. Now, why is that? Because influencers, along with other viruses, coronaviruses, such as the one that we're having right now, they are RNA viruses. Okay, RNA viruses. So for PCR reactions, you go from DNA to DNA. Okay, for RNA, there is a little small steps. Okay, we're going to talk about it in the next slide. But the idea behind these diagnosis is to gather some of the respiratory specimens, and then you perform basically, very crudely, you can do some purifications and then do a PCR reactions with the purified specimens. These tests are usually very highly sensitive and specific. So and in some cases they can tell differences between influenza A and B and different subtypes, basically where H5N1, those subtypes. Okay, you may have H3N2, these, or swine flu, those subtypes of influencers. Right. The time it takes for these reactions, it ranges from as fast as 45 minutes to several hours, depending on the particular gene that you need to amplify. Okay, and also depending on the specimen samples, as well as the machines. Okay, if you have a better machine, you do quicker, better thermocycler. Okay. Now, what is RT-PCR? Okay, RT-PCR, it's just one step before the real PCR. It's so-called the reverse transcription steps. Okay, you start with a viral RNA. Okay, again, you have a primer to make a cDNA. Okay, copy of the DNA, so that now you have the first cDNA. And then after this reverse transcription step, you're going back to the regular PCR. Basically, you have a DNA starting material, and then later on amplify and amplify and amplify, just like your regular PCR. So, like I said, there's only this small difference at the beginning there. Alright, graphically, okay, actually there are two steps, okay, two steps RT-PCR, and one step here. Now, the one step is you have a sequence-specific primers mixed with your messenger RNA, or that is isolated from the specimen sample, and then you put in reverse transcriptase, DNA polymerase, and all the DNTP, the nucleotide building blocks, into one tube and perform it in one pot. Or you can do two-step, i.e. you do your reverse transcription first, alright, and then you do your obtain your cDNA and then do your PCR. So there's virtually no difference. Of course, one step is easier, quicker, but usually one step they are a little bit more expensive. Like anything, more convenient, more expensive. I've done a couple of those more recently in one of a side project there. The one step is really nice. Yeah, I go for one step. Alright, so now there is a small variations between these RT-PCR, so-called real time. Okay, now usually RT-PCR, people get confused. I sometimes get confused myself. Are you referring to reverse transcriptions or real time, right? So you should, technically RT-PCR should be referring to as reverse transcriptions. So what it means is going from RNA. Oh, sorry, very, very bad handwriting. RNA to DNA. Right, DNA. And real time, it's real time. Okay, basically you're knowing what is happening. And then there is an endpoint. So at the end, we're looking everything together. The steps are very similar. So here we have RNA isolations and analysis, making first strands of cDNA. Now, in the real-time PCR amplification, there is a usage of a fluorescent measurement so that you can see strands are being produced are being produced in a real time, and it's a little bit more fancier machine that pharmacy doesn't have it, but in biology department, in the Cohen Science Building, we do have a RT-PCR machine where you can utilize fluorescent molecules to get an idea of how much DNA are being produced every cycle. Basically it's a quantitative measurement. Okay, it's a quantitative RT-PCR. So sometimes people call it like that. And then endpoint. The endpoint is old school in a way that you perform your PCR, amplify to a certain amount, go through DNA gel electrophoresis, like the gel that I show you in the first slide there, and then looking at the gel with a imager, capture an image, see if there's a DNA in all those. Now, obviously this is not as sensitive. Okay, the gel image, it need to be at certain concentration in order to show. Many times in my lab, students sometimes are not sure if the certain DNA pieces are not, is it amplified or not, because it comes with experience. Sometimes I see a band, students don't see a band, but after they've been in a lab for a while, they start developing the very keen eyesight, the eyesight of an eagle, that can spot a very faint band on a particular gel that they weren't able to when they first start. But so it is, it's very user dependent. But that's what I mean. It takes time to get to that skill. But versus real time, even though the setup is a little bit difficult, but the computer with computerizations and the instrument help, you can tell the data, the analysis is much nicer. I personally have done more of the endpoint versus compared to real time. So that, because I don't have a real need to at this point, even at this point in time. So the advantage and disadvantage of PCR, okay, when we're talking about PCR in general, is that advantage, they are highly sensitive, relatively easy to set up. We're talking about like 5 or 6 basic ingredients, putting it together and then pop it into the thermocycler. I call it oven, and then you bake it and then you come out, right? It's relatively easy and relatively fast turnaround time. It could be, on average an hour or 2. You can get something out of it, particularly for virals. It's very difficult to culture viruses in your dish, right? Unlike bacteria, where you can do bacterial culture and then do some tests and see, okay, that is this bacteria, like Pseudomonas versus a pneumococcal, whatever that you're looking at. Viruses, on the other hand, it's difficult. You can see it. So PCR, it's almost like the gold standard for the high sensitivity, at least before any better assays were being identified. So that is the advantage. Disadvantage of PCR in general is liable for contaminations, particularly when you are trying to when you're talking about isolating mRNA or genomic RNA from an organism, because RNA by nature, they are very, very easily being degraded. So when I do RNA extractions, I usually need to clean the bench with like bleach and 70% alcohol and wipe it out, tape it off, like off limit. This is for RNA only, because there are RNAs everywhere. Okay, it will degrade your RNA very easily, and that is what it means by contamination. So you lose the starting product already before you do anything else. Then that leads to false negative, right? So that is a thing, and that is a concern. And also tied to operator skills, okay? Operator skills. Students in my lab started off doing PCR. The first 1 or 2 maybe not so successful, even though I was doing along with them. And the idea behind it is that it needs practice, okay? After a while, people get good at it, but still, that is it requires some steps involved. And then also the main thing is, having a presence of a particular pathogen causative, a viruses, for example, doesn't necessarily means you have an active infections. Some viruses are latent, okay, you can detect it, the presence, but you're not actively sick at certain points. So that is another advantage of PCR in general. Alright, and then here we are going to talk about the assignments, and the assignment here is to research and look for a PCR based kit that is used for COVID-19 diagnosis. Okay, COVID-19 diagnosis. So you hop on the internet, okay, do a little bit search. Okay, the search term could be as easy as COVID-19 PCR. Alright, so and then it will pop you a lot of websites. You're going to look for one, okay, only one product, okay, that is marketed for COVID-19 diagnosis, right? So and then you answer these following questions, okay, and things like what company is making it. A lot of these looks long, but you're talking about one word. What company? Eurofins, Cryogen, whatever. Okay, whatever you can find. Those are one word type of answer. Okay, I'm not expecting like crazy write up in this assignment, right? This is due in 1 week, and it's a nice, quick, easy thing to do, I believe. And then you go to those websites, look for they usually have package insert or product related informations, looking at their advertised sensitivity and specificity, right? How much viral load they can detect, okay, and how fast is the reaction? How much time do they advertise from isolations to a read to a data analysis? How fast is that reactions? And also, now notice a lot of these kits, some are approved, FDA approved for diagnosing purposes. Some are only for research purposes. Okay, whatever you can find is whatever you can find, but be able to realize, is this for diagnosing, real diagnosis that can be used as in a hospital lab, and tell people, hey, you have this disease, versus research purposes, something that Dr. Hong will use, in his lab doing this. And while I can say you are highly suspected for this infection, but I cannot certify that you are infected. So diagnosis versus research. Right, is it a endpoint and versus a RT-PCR? Okay, we just talked about the differences between real time and endpoint. So again, it is just spotting those informations. And then the rest of the questions, 6 and 7, depend. Now, if it doesn't have it, it doesn't have it. Don't stress it. Does the kit provide information on the primer sequences? If so, please let. I doubt, but I'm curious. Okay, I'm curious if people are telling people what primer sequences they are including in those kits. Does the kit provide information on specific genes that they are amplifying? Okay, and then these are more technical things, may not be easy to find. Okay, don't struggle too much. If so, please list the informations. Okay, these are not gonna be, if you can't find it or they don't have it, it's not gonna count against you in terms of the point. Okay, and then lastly, copy and paste. Not lastly, and then last questions. Copy and paste the websites and diagnosing kits in the documents that you prepare, so that I can click to it and then look. Okay, what is in there. And then finally, reflection. Now, a lot of you may be finding very similar, same kit. That's okay. I expect that. And those answers basically are not going to be varied too much. Now, the last one, okay, reflections, okay, in your opinion, okay, this is an opinionated questions, really to keep and makes you think a little bit. What is the major advantage or disadvantage of the product that you find? Okay, not about the price. Someone will tell you this thing is a box of kit is $120 and it can process 30 samples. We're not talking about price. Okay, we're talking about other factors that you think it's good about this kit or not good about this kit, and then you will write a small paragraph or so, a couple sentences to express your opinion there. So I expect this opinion, the questions would be different, right? Quite different from each one of you. Right? If I see something that very similar between the two of you, that indicated of something potentially, you guys are exchanging mind. All right. Okay. So here is the picture on the side is the SARS-CoV 2 structure. And yeah, it's just showing this is a RNA viruses. Okay, I'm not gonna elaborate too much ssRNA here. Okay, I'm not gonna elaborate too much in terms of the viral structure and all these. All right, so that is for this week. School closure week 3. And good luck with the assignments, and let me know, email me and things like that. Leave in the discussion board if you have any burning questions on what to expect. All right, and I will see you next week.